Every living organism uses the exact same genetic code. Every organism stores genetic information in the 4-letter DNA alphabet. And every organism uses RNA, messenger RNA, to carry that gene message. Transfer RNA to link up genetic information to amino acids, and finally ribosomal RNA to assemble the proteins. The exact same genetic code is used by every organism, so the human genes can be inserted into any other life form. In the past half-century, molecular biologists have learned to read that code, and they have come to recognize that every individual has a unique genome. In this lecture, I want to tell you about 2 dramatic developments related to our newfound understanding of the genetic code and its application to the study of humans. First, I'm going to focus on genetic fingerprinting and genetic testing. These are technologies facilitated by the polymerase chain reaction, or PCR. Then I'm going to consider the extremely controversial and ethically challenging field of behavioral genetics. As we begin this lecture on each individual's unique genetic heritage, let's turn our attention back to the human genome. That's an incomprehensibly complex array of some 80,000 genes distributed on 23 pairs of chromosomes. The power of molecular genetics lies in its potential that it can affect everyone's life by understanding this unique genetic code that we each carry with us. Scientists now have the ability to differentiate the unique genetic pattern of almost every human individual, often on the basis of DNA contained in just a few cells. Genetic fingerprinting makes it possible to identify an individual from the genetic material in just a single cell. Now, single techniques have begun to tell us a person's genetic predispositions as well, perhaps months before they're even born. All humans have the same set of genes. They're arranged in the same way on our 23 pairs of chromosomes. But among these 3 billion base pairs, millions of individual differences exist between any 2 of us. Typically, once in every few hundred, maybe once in every 1,000 base pairs, there will be a difference, and that's what makes us unique. Now, some of these differences are insignificant. They're hardly noticed because they're just noise in the background of genetic information. But other differences are responsible for things like inherited diseases, things that can change your life, things that can lead to premature death or debilitating disease. We are all different. None of us is alike, and that is because of these differences in the genetic code. DNA fingerprinting, which makes dramatic headlines in White House sex scandals and lurid rape trials and murder trials and so forth, it's now used routinely to set free the innocent. It's used to convict the guilty. Let's see how this process works. Let's also look at genetic tests that can expose the most intimate secrets by providing incontrovertible evidence of contested paternity, of family lineage. You even see advertised in newspapers these days, 1-800-WE-DO-DNA, advertising the idea that you actually can go out and test in a commercial way genetic information that a few years ago could never have been done. How is it possible? How have we made these genetic advances? As we're going to see in 2 lectures, detection of certain gene mutations is a reliable indicator of some hereditary diseases, inherited diseases. In other instances, detection of mutant genes can, for now, only indicate an increased risk of a certain kind of disease, but that's important too for medical diagnosis. Our ability to manipulate and to analyze tiny amounts of DNA rests on a simple and elegant technique. It's called the polymerase chain reaction, or PCR for short. This relies on DNA's ability to duplicate itself. PCR targets a specific short segment of DNA, and then it makes countless copies of that segment and that segment alone. Here's how it works. You begin by preparing a solution that contains the DNA sample of interest. Perhaps it's the chromosomes from a single cell. Now, the target segment of DNA is often a very, very tiny proportion of that total, maybe only a few dozen base pairs long. To the solution that contains that DNA, you add large amounts of 3 key ingredients. First of all, you add individual DNA nucleotides. That's the individual rungs of a DNA ladder, the A, the C, the G, and the T, so you can make lots of copies because you need those raw materials to do it. Second, you add an enzyme called polymerase, and that helps to assemble DNA strands from nucleotides. That's what all enzymes do. They help you build up to perform chemical operations and speed them up, and polymerase speeds up the formation of DNA. And finally, you have numerous copies of what are called short primer segments of DNA. These segments target and recognize and bond to only specific desired sequences of perhaps 20 base pairs long on specific DNA areas, specific chromosomes. So out of the whole 3 billion base pairs in human DNA, for example, you might be able to target one short segment and just duplicate that segment over and over again. Well, this solution is heated to about 200 degrees Fahrenheit. That's just below the boiling point of water. And at that temperature, the DNA double helix automatically splits apart into single strands. It doesn't happen in nature because we don't normally get to 200 degrees Fahrenheit, but in a solution you can do this. So where before you had double helices, now you have 2 single strands. Now this mixture, which is separated, has single strands of DNA. It's cooled then to 140 degrees Fahrenheit, and that's cool enough for 2 of the primer strands to bind to the very specific target segments on the 2 exposed DNA chains in the target gene. So if you can imagine, you have 2 separate strands of DNA, and 2 very short segments now become a double helix in an otherwise single-stranded molecule. So polymerase triggers the growth of new DNA strands from that point, and you start making new segments from those primer spots. The result is 2 new double helices of the short segment, where before there was 1. And then you repeat the process. You heat back up to 200 degrees. The 2 short segments of DNA that you formed, the 2 new segments, split apart, and now you have 4 exposed primer pairs. You cool it down. Then you get 4 new double helices. You heat it up, you have 8 single strands. You cool it down, you get 8 double helices, and so forth and so on. And each time you cycle the temperature, you increase by a factor of 2 the number of strands. And within a few hours, you can have millions of copies of the targeted sequence, while the rest of the cell's genome remains in its original single strands, the 2 single strands that you started with. Now, given this large amount of a single gene or a single segment of DNA, it's often possible to just use routine procedures to determine the specific DNA sequence of that particular segment. This polymerase chain reaction has found many, many applications in science and in technology. The most publicized uses are certainly in the newspaper. They're the forensic applications where you use it to identify small amounts of DNA associated with a crime scene. If you have traces of blood, a few cells of skin, perhaps some semen, or other cellular material, you can amplify specific segments of that DNA and look to see the characteristics of that particular individual. You can then take a similar blood sample from the suspected crime person, or perhaps a victim, if you don't know who the victim is, and you can do the same sort of test, and you can match up these characteristic sequences to see how close they are. If they're identical, then you have a match. The United States is now following the lead of many foreign countries in stating that DNA test is a basis for convicting people, and they're also beginning to think about starting a DNA database for sex offenders, an idea which has many controversial ethical implications, but support on both sides. It's an interesting issue to debate with other people how you feel about that. Police and military experts also have used DNA fingerprinting to identify casualties of war, victims of bomb blasts, for example, and other unrecognizable human remains. It's really quite a sobering thought to me, who grew up with the Tomb of the Unknown Soldier, that never again are we going to have an unknown soldier. Since everyone who goes in the military has a genetic fingerprint made, any remains that are recovered will be identified according to this technique. So the Tomb of the Unknown Soldier is now kind of an anachronism. It's sort of sad in a way because it was a great symbol. PCR provides the molecular biologist with a powerful tool to identify and to understand genetic characteristics. It becomes a research tool in the hand of the molecular biologist. It's found routine use as a rapid screening technique for numerous genetic diseases also, such as cystic fibrosis, muscular dystrophy, for viral contaminants, such as HIV in blood, for tissue compatibility before organ transplants. Anytime you're trying to compare and contrast two pieces of genetic material, PCR provides a wonderful way of doing this quickly, cheaply, and efficiently. PCR amplification of viral and bacterial genes, which, remember, mutate very rapidly into new variants, is extremely important now in disease control. You track the spread of different strains of viruses, different types of bacteria in this way by recording their unique signatures, and once again use PCR to amplify the genes. This technique is also playing a very important role in understanding how a gene works. It documents the protein structure for which it codes, and this is a fairly tricky process, so let me try to explain this to you. This uses that reverse transcriptase I told you about in the last lecture. Now, the first step is to find the location and the sequence on a strand of DNA. In some cases, the DNA sequence exactly matches the protein. That is, every triplet of bases matches one amino acid in the protein. In such a case, determining the protein structure is straightforward. But remember that in many cases, the structure of a gene is very complex. You may have breaks in the sequence. You may have these things called introns. So you have to have a variety of editing instructions in the original chromosome to get the messenger RNA and then to make the protein. So just reading the sequence of letters in DNA isn't enough. You have to know the editing instructions. Now, cellular mechanisms have to remove these intervening sequences, and they have to splice the complete gene together before you can make a correct messenger RNA. That's something the cell does very efficiently, but something that we don't know how to do yet. Since we don't know how to do it, we have to trick the cell into doing it in a different way. And here's how it's done. You actually can take an RNA, a messenger RNA strand, that's all set to make the protein, and use that and reverse transcriptase to go back and make the DNA. And then you can use the polymerase chain reaction to duplicate that. Think about how this process works. You let the cell make your messenger RNA for you, because the cell knows how to do it even if we don't. Then you can take the messenger RNA, you can apply this special protein, reverse transcriptase, which does that weird thing of converting a messenger RNA back into DNA, something that viruses are very good at. And now you have a complete strand of DNA that exactly matches the gene. It doesn't have any of that interfering intron material in it. You multiply that with polymerase chain reaction, and then you can sequence the DNA and determine the sequence of amino acids. And that can then tell you something about the structure and the function of the protein. Well, I'm going to switch gears now and talk about one of the greatest ethical challenges that's been spawned by our growing understanding of the human genome. This newfound ability to read an individual's unique genetic heritage and our growing understanding of the specific function of various genes leads inevitably to the kind of ethical questions about how we control, manipulate, store this information, who gets to see it, and so forth. We certainly can admit that each person's interests and abilities and behavior arise from a very complex intermix of environment, background, and also genetic attributes. It's undeniable that genetic factors play a role in who we are and what we do. You can't get away from that. Some of these factors are obvious. For example, there's some genetic diseases that are invariably fatal by a certain age. But there are other traits such as height, musical ability, sex drive, which are much more subtle, and how much those are environmental concerns and our background and experience when we were growing up, as opposed to specific genetic controls, it's very, very hard to say. Here's a quote from molecular biologist Torsten Wiesel. He said, quote, "Perhaps most disturbing to our sense of being free individuals capable to a large degree of shaping our character and minds is the idea that our behavior, mental abilities, and mental health can be determined or destroyed by a segment of DNA." Well, like it or not, there exist incontrovertible links between human behavior and genes. These links were discovered essentially by accident in studies of drugs and studies of illness, other seemingly non-behavioral phenomena. For example, there's a defective gene that blocks hunger suppression. That can lead to overeating. You've perhaps known a child that has Down syndrome. That's a genetic disease. But these children are exceptionally kind and gentle. On the other hand, another genetic disease, Tourette's syndrome, may lead to people who display extreme antisocial behavior. So in the case of these genetic diseases, there's a clear link between a gene and a behavior. Now, equally important is recognition of the familiar concept of one gene, one disorder, that defects in single genes are necessary and sufficient to cause specific diseases, that you can't extend that to behavioral traits. Behavioral traits are much more complicated. They are the result, at best, of a large concatenation of many genes as well as environmental characteristics. There's not a single gene for people who are happy, or a single gene for a musician, or a single crime gene, certainly. But rather, there's complex behavior and abilities that arise from this interaction of a product of a whole collection of genes and also, as I said, many environmental factors. We certainly don't know all those genes and all those environmental factors. So this research takes on a rather difficult turn. There are daunting ethical problems in studying human behavior. And so, at least for the time, animals provide an ideal means to study behavior without raising many of those difficult social and political questions that are associated with the human behavioral research. We have centuries of observations on selective breeding of domestic animals, and these demonstrate beyond any reasonable doubt that behavior is determined in part by heredity. Selective crossbreeding has led to great gentleness in some domestic dogs. It's led to aggression in killer bees. You all know that different breeds of dogs have different degrees of aggression, and that clearly is a genetic part of their makeup. There were pioneering studies that were begun in the 1960s to understand some of these genetic relationships to behavior. Seymour Benser of Caltech was zapping fruit flies. He used radiation, he used mutagenic chemicals, and he watched for mutant offspring. These are offspring that displayed unusual behavior. Now, most flies are unaffected by this radiation, but a few would fly or feed or rest, or they'd mate in very odd, strange ways. Benser and others have subsequently identified numerous genes that participate in the routine functioning then of the fly's nervous system and in its behavior. You can do the same kind of studies with mammals, but mammals display far more complex behavior. They're much less amenable to comprehensive study than flies, partly because it takes a long time to go from one generation to the next. Flies can reproduce in a matter of a few weeks, as opposed to mammals, which take months or years. But of course, mice are much more likely to shed light on hereditary influences in humans, and so they are the study of intensive research. In one widely used experimental protocol, biologists crossbred two strains of animals that display contrasting behavior. Purebred, highly aggressive mice are crossed then with docile mice, for example. There was a team of British and American scientists led by the behavioral geneticist Jonathan Flint, who's basically used this protocol to identify DNA segments that are found primarily in unusually fearful mice. This is an amazing sort of strategy. You have to decide how aggressive or fearful a mouse is, and this is how they do it. Since mice display a wide range of behavior, you put a mouse into a brightly lit open area. You'll see that some mice actively explore their new surroundings, running all over, just sniffing around, while others cower in one corner, and this is a quote, while defecating copiously. Those are the fearful mice. So you can measure this wide range of behavior and give a scale perhaps of 1 to 10 of a qualitative estimate of how aggressive or fearful a particular mouse happens to be. What Flint and co-workers found was a high degree of correlation between fearfulness and 3 specific DNA segments. And then you have studies of what are called knockout mice. These are mice in which researchers specifically inactivate a gene that then can reveal a clear link between mouse behavior and that particular gene. In 1993, a team of scientists led by the French microbiologist René Hen targeted a gene that activates specific neuroreceptors for the chemical serotonin. This transmits signals in both mice and in human beings. Now, these knockout mice displayed extreme aggression, leading the French scientists to speculate that similar defects might contribute to abnormal human behavior, extreme aggression in humans. Well, as I said, human subjects are vastly more difficult to study than mice. And I think for a time, any controversial conclusions about heredity and human behavior are going to be the subject of scathing attacks on grounds of questionable experimental methods, such as sample size or lack of sufficiently rigorous controls. And it's interesting, people on any side of the political spectrum can argue this way. For example, if you wish to ignore the conclusions for ideological reasons, you can rationalize a choice on scientific grounds. The recent debate over genetic links to homosexuality based on molecular genetics, this is research by Dean Hammer of the National Cancer Institute, provides a wonderful and widely publicized case in point. Hammer claims that there's genetic basis for homosexuality, but others who don't like to hear that sort of rhetoric will denounce it, saying his studies are flawed. If you're going to do behavioral genetics, you have to make 3 critical assumptions. And so rather than debating whether or not any specific case is flawed, think about these 3 assumptions and see if you agree with them. The first assumption is that behavioral and personality traits can be ranked, at least in some sort of qualitative way. For example, behavioral researchers generally agree that personality rests on 5 broad traits. And here they are. The first is called extraversion. That's whether you're outgoing or decisive or persuasive, for example. The second trait, according to behavioral geneticists, is neuroticism. It includes the traits of being emotional, irritable, or anxious. The third trait is conscientiousness. That's whether you're organized, practical, dependable. Then there comes agreeableness. Are you good-natured? Are you warm? Are you kind? And finally, the 5th trait is openness, curiousness, insightfulness, imagination. To one extent or another, each of us displays a combination of these 5 traits. And the assumption is made that you can have some sort of qualitative ranking of individuals, sort of a 1 to 10 scale of people, you know, how they fit into each of those 5 characteristics. It's assumed that similar rankings are possible for different kinds of intelligence. Music ability, for example, memory, spatial reasoning, verbal ability, for example. Although I think all people agree that a single number for IQ is meaningless. Okay, that's the first assumption, that you can rank these characteristics. The second assumption of behavioral genetics is that environmental factors influencing behavior can also be identified. Now, there are many of these factors, and it's difficult to identify them all. They can include things like nutrition, exposure to drugs, sleep patterns, stress. Those are all well known to affect behavior. But there are also other influences that may be quite subtle, such as visual stimuli when you're in infancy, pollutants in air and water. Exposure to sunlight could be one. All types of physical activity. And these may be much more subtle and difficult to track down in trying to figure out relationships to behavior. And then there's a third assumption. That is that distinctive genetic characteristics of individuals can be quantified. And I think that's unambiguously true. Whether we can make a correlation with behavior is not so necessarily true. But the day is going to come when the entire human genome can be sequenced automatically, perhaps in a matter of a few days. So rather than having a single human genome, which is what's envisioned by the Human Genome Project, we're going to have access to thousands of different human genomes of individuals. And each of those is a detailed chemical map of a unique individual. So here we have these 3 assumptions: that you can quantify behavior, second, that you can identify environmental factors, and third, that you can determine genetic factors. Now, if all 3 assumptions of behavioral genetics are realized, then you can perform statistical tests, and they may be able to establish definitive links between heredity and certain kinds of personal traits. You could, for example, imagine that you do the genomes of 100 gifted musicians, and maybe 100 people who express that they're tone deaf, they can't even carry a tune. And you might then see that there are certain combinations of genes that are always present in the musicians and not present in the people who are tone deaf. I don't know if that's going to happen or not, but the fact is, in 10 years or 20 years or 50 years, we will be able to do a test like that, and some scientists will. And perhaps we'll be able to identify musically gifted children at birth. Whether we want to do that is a different question. Scientists and the public at large are strongly divided over these ethical implications of genetic testing. You see, proponents see this research as the best hope for understanding and ameliorating antisocial behavior. If genetic traits influence behavior, then improving a child's environment represents our only realistic hope of helping out the children. You can't change their genes, at least, well, maybe you can't, but changing the environment is something society can do, and maybe that's our responsibility. If, on the other hand, one adopts a more conservative agenda, then findings of behavioral genetics might be equally assuring. What you might say is that genetic traits absolve the society of responsibility for things like poverty, for crime, for drug abuse, so society can't take responsibility because it's an individual genetic thing. And so it's not an environmental question, they will say. I think by far, and I'm sure you would agree, that genetic racism becomes the scariest of the scenarios here. That if we can in fact identify certain genes and then discriminate against people because they carry or they don't carry a certain gene, that becomes the scariest possibility of all. Behavioral genetics forces us to confront the fact that we are, in essence, chemical beings. We are vastly complex. We're adaptable, we're unique. We're unpredictable, to be sure, but we're still chemical beings, and those chemicals are determined by our genes. We're learning to identify those chemicals. We're learning to decipher how they're made. We're learning to discover what they do, and even how to fix them when they're defective. In the process, science, once again, has forever changed the way we think about ourselves and about our place in the universe. I want you to imagine a time when every genetic predisposition of every individual can be known. When we have aspects of health, personality, intelligence, behavior, they all can be quantified before birth because we can extract a single cell from an embryo, we can do a complete genome on it, and we already have all of these tests where we know the characteristics that lead to various behaviors, various abilities, various potential medical problems. Who should have access to that genetic information? Should you? Should your parents? Should your doctor? Should your employer be allowed to see it? What about your health insurance company? Should they be able to see those records? What about the government? What happens in a day when everyone in military service has their genome taken and the government knows that information whether you want it or not? Should there be any policies of any sort relying on how this information is distributed? Remember, all this information can be gleaned from a single cell, perhaps without your knowledge. Well, to summarize, we've come to a point in human history when humans can identify any individual from his or her unique genome, the genetic material of a single cell. Using the polymerase chain reaction, or PCR, you can take DNA from that single cell and amplify it and analyze it. Then this information can be used in a whole variety of ways. You can use it for DNA fingerprinting, for identifying criminals or victims from the DNA of a single cell or groups of cells. You can use it for genetic testing, perhaps looking for specific markers for diseases or other genetic traits and other markers. You can use it for family testing, for trying to determine the mother and the father of an infant, or perhaps other family lineages, as in the recent case of Thomas Jefferson and Sally Hemings, trying to see if they in fact shared the parentage of children. And now we have discoveries in the field of behavioral genetics. And we someday may be able to predict the character traits, the abilities, potentially the flaws of children before they're even born. And as a society, we have to make a decision on how we're going to use that information. But I think the biggest ethical dilemma of all is going to come when, in the very near future, we learn to fix defective genes. And that is the subject of the next lecture.